Okay, so today we're going to discuss all about the bacterial genetics. Okay, so before we proceed with one, so we have here the basic structure of the DNA or the nucleotides. So again, as a review, your nucleotides made up of number one, nitrogenous bases, and they are having the base pairing. So again, your AT, adenine, try to base pair with your thymine in the case of your DNA. But in the case of your RNA, your thymine is being replaced by the uracil, which try to base pair with your adenine. Another one, we have your GC guanine, try to base pair with your cytosine. Okay, the next component of your nucleotide, we have here your pentose sugar, 5-carbon sugar. Okay, so in the case of your DNA, that one is deoxyribose. Pag sinabing deoxyribose, so wala siyang oxygen. Okay, unlike with the RNA, so that one is ribose, meron siyang, this one would have here, may hydrogen, ito ay may OH. Oxygen saka hydrogen, this one, may naka-attach sa kanyang 5-carbon sugar na only the hydrogen. So this one, wala siyang oxygen, that's why tinawag siyang deoxyribose. Another component of your nucleotide, we have here your phosphate group. And for the strand, the RNA, we describe this one as a single-stranded. The DNA, on the other hand, we describe this one as your double-stranded. Okay, then we have here other terms. Okay, the first one, we have here your so-called here. Okay, we have here the term gene. So gene here is made up of the different DNA sequence. Okay, that try to comprise, okay, the entire chromosome. Okay, the gene here is made up of the DNA sequence, and each of the DNA try to code here for, is your gene, try to code here for a genetic information that you need to transcribe and translate in order for that to produce a particular product. Sample the product, we have here your protein. Okay, the genome on the other hand is made up of the different set of genes. So malaki ang genome natin compared to your gene. Okay, ulit, pinakamaliit is your DNA. Then, madaming DNA that become your gene. Madaming gene that become your genome. Your genome, again, could be in the form of your chromosome, or it could also be in the form of your extra chromosomal elements. So, mean to say in the bacterial cytoplasm, again, the chromosome of your bacterial cytoplasm here, we describe that one as, ano ang chromosome nito? Singular, circular, and that one is a double-stranded. Pero very simple lang na chromosome. Extra chromosomal elements, so this could also be found here in the cytoplasm. Aside from the chromosome, may makikita ka pa na another circular, single circular exochromosomal element. We're talking about the plasmid. Okay, so again, we describe here the chromosome contains all the genes which are necessary for the growth and replication of your bacteria. And for the bacterial chromosome, we describe that one as simple, circular, double-stranded. Okay, then we have here the term mobile genes, or you call that one as your jumping genes. So these are from the word itself, mobile, so jumping, these are the genes which could eventually jump from one area to another location within your chromosome. Example of this mobile gene or your jumping gene, we have your IS, insertion sequence. Okay, insertion sequence, consider here to be the simplest piece or simple piece of the DNA where it could eventually be break here from one in and out of your DNA. So, makatransfer siya from one DNA to another, and therefore from one area in your chromosome to another. Okay, your insertion sequence, sometimes here once it try to migrate, okay, or jump from an area to another area, especially if it jump here at the middle portion of your chromosome, it might disrupt, okay, the entire property of your bacteria. Like for example, its ability to ferment the carbohydrates. It's because again here, okay, nag-transfer siya from one area to another, so nagkakaroon ng mutation. And it might eventually, yung pinaka-effect niyan, it might disrupt here the entire property of your bacteria, including, for example, its ability to ferment the carbohydrates or other properties. Okay, another example of your jumping gene or your mobile gene, we have your transposon. Okay, the transposon here is a jumping gene, most likely could be found here within your plasmid. And therefore, this one try to confer also here antibiotic resistance, especially when that one is being exchanged between two bacteria. Then we have your extra chromosomal gene in the form of your extra chromosomal element in the form of your plasmid. Again, the plasmid is also circular double-stranded DNA that could also be found here in the cytoplasm, aside from the chromosome of your bacteria. Okay, the plasmid here contains the genes which could confer here resistance against your antibiotic. And this plasmid could be exchanged between your bacteria. And therefore, one bacteria which is resistant to your antibiotic, and another one is not resistant to antibiotic. If the plasmid of that bacteria which is resistant to antibiotic is being transferred, okay, to the another bacteria, wherein that one is not previously resistant to antibiotic here. However, since the plasmid of the another bacteria which confers resistant antibiotic has been taken up by that bacteria na previously hindi siya antibiotic resistance, since na-take up niya, it would eventually here confer resistance na rin. Because again, the plasmid is a way here of genetic exchange among your bacteria by the process called here your conjugation process. Another one, the plasmid could also encode here for or code here for the gene that would confer here toxin or toxin producing the gene. And that again, this toxin here is responsible for the virulence of your bacteria. Sabing virulence, the more virulent it is, the more pathogenic it is. And that could also be exchanged from your bacteria to another bacteria. And that's your extra chromosomal element in the form of your plasmid. Okay, we're going to discuss a simple discussion about your central dogma. Okay, the first one, we have your DNA replication. So DNA replication is simply as, just the copying of your strand of your DNA. That's basically to increase the number or increase the copies of your DNA. Your DNA, again, contains here the genetic information, and that genetic information code here for a production of a particular product, like for example, the protein. So as our view, we have here DNA replication starts with the unwinding of your double-stranded DNA, by the enzyme helicase. And you could also have here your topoisomerase to prevent the reformation of your double-stranded. Pag na-unwind na siya, what you have here are two strands. One is your leading strand, the other one is your lagging strand. So again, the leading strand will have your 5' to 3' direction, and the leading strand is 3' to 5' direction. In each of this strand will be copied here by your DNA polymerase in order for you to have another copy of your DNA. And therefore, what you have here are more copies of your DNA. Each of that contains here a genetic information. Again, genetic information code here for production of particular product like your protein. Okay, the next step, we have your transcription. Transcription is just transcribing or you just transferring the genetic information from your DNA to your messenger RNA. And you'll be needing the enzyme RNA polymerase for that. Okay, so your replication and transcription are occurring within the nucleus. Okay, within here the cytoplasm, because you don't have the nucleus, but that's still within the chromosome of your bacteria. Translation process, on the other hand, occur here on the cytoplasm. Okay, sa labas na siya. Translation process is primarily the formation here of the different substances which are needed for the translation. We have your messenger RNA, contains here the codon. Kung sinabing codon are triplets of your nucleotide base. So like for example, we have your triplet, we have adenine, uracil, guanine. So this is your codon, for example. Another one, this would eventually form a complex along with your ribosomal RNA. That's your small and large subunit ribosomal RNA. Plus, we have also here the transfer RNA. The transfer RNA try to carry the anticodon. So, anticodon will always have ano lang, kung opposite lang nito ng base pairing natin. Kung itong codon natin, ang anticodon mo for that A would be your uracil, this is A, then C, ganyan. This is your codon, this is your anticodon. They still have here their base pairing. In order for that to be translated to become your amino acid. Okay, so the synthesis of your protein or your amino acid starts here with your start codon. Pag nakita ng start codon, along with your anticodon, it eventually tries to synthesize that one. Your start codon is your AUG, that's your methionine. And magsa-stop lang siya synthesizing here your amino acid if it try to reach here or try to find here one of the stop codon. One of your stop codons are the following: UAA, UAG, and UGA. So pag nakita niyo ito, it will stop synthesizing that one. And after that one, if you have an individual na mga amino acid, you try to package that one, you try to assemble that one to form here a bigger complex protein structure, and that become the product here for your particular replication, transcription, and translation process. Okay, the next one, we have here your genetic alteration process. Okay, we have here the mutation. So mutation is just an alteration in the usual DNA sequence. Okay, so the effect here of the mutation, since nag-change ang sequence ng DNA within your chromosome, for example here, so it might eventually affects also here the synthesis of the protein. It might eventually could not synthesize the protein anymore here because of the alteration and the supposed to be the normal DNA sequence. So we have here different types of the mutation. The first one, we have here the point mutation, or you call that one as your base substitution. So this is just only characterized here by only one alteration in only one DNA or nucleotide sequence. So instead dapat na adenine doon sa position na yon, pinalitan siya ng isang nucleotide na hindi dapat yon ang nucleotide in that position. You call it one as your point mutation or your base substitution. On the other hand, we have also the frame shift mutation. Okay, the frame shift mutation here is a type of alteration characterized by changes in more than or two or more na nucleotide sequence. Okay, the next one, we have your genetic recombination is the process whereby the DNA is being recombined or being re-exchanged or exchanged among homologous chromosome here of your two or more bacteria. So one chromosome here coming from this bacteria, it try to exchange to another bacteria. So becoming part of its own chromosome, you call it one as your genetic recombination. Okay, we have here the different process of your genetic recombination. The first one, we have here the transformation. Okay, the transformation is just the process whereby your naked DNA in the environment is being taken up by your bacteria. So, that naked DNA, pwede siya galing sa bacteria na namatay na, where it try to release the DNA in the environment. So, pag nakita ito ni another bacteria, it could take it up, and you call the process here as your transformation. Once the bacteria try to take up the naked DNA from the environment, pag pumasok sa katawan ni bacteria ang kanyang naked DNA na take up niya, it try to recombine with its own chromosome. Pag nag-combine na siya, it eventually try to undergo the process of replication, transcription, translation, and becoming part of its own chromosome niya. And remember that naked DNA here might code here for particular property na hindi common here or hindi natural na property ng bacteria. But since it try to take up that naked DNA, it might eventually try to influence okay, the production of the new product of that bacteria will try to take up that new DNA from the environment. So there are some bacteria, there are only few bacteria which are capable of taking up the DNA in the environment in a natural process. You call that bacteria here as a competent na bacteria. That includes your Streptococcus pneumoniae, Haemophilus influenzae, and we have your Neisseria gonorrhea. In the laboratory, however, we could also induce the transformation process here by your cloning process. So like for example, the gene here coming from E. coli, ay pwede ka mag-insert ng naked DNA or foreign DNA from your E. coli by the process of your transformation. Okay, the next process of your genetic recombination, we have here your transduction. Okay, the genetic recombination through your transduction process is basically mediated here by your bacteriophage. So bacteriophage is just your phage. We try to infect your bacteria. The phage itself is another organism. Primarily contains the chromosome niya. Contains here either the DNA or the RNA. So itong chromosome niya, RNA or DNA. Then meron siyang sariling protein coat which try to surround the chromosome. Again, your transduction process here is basically process of genetic recombination through your bacteriophage. Okay, then we have here two types of your bacteriophage. One is the lytic phage. Another one, we have your lysogenic phage. These are actually the life cycle of your bacteriophage. So the first one, we have here your lytic phage. So basically, the procedure here for the infection here caused by your phage. So first, we have your phage here try to infect your bacteria. As it try to infect, it try to introduce its chromosome. Remember here, the chromosome of your phage could either be a DNA or the RNA. I try to infect the bacteria. It try to leave primarily the protein code here outside. What is being introduced only inside your bacteria would be the chromosome. So once introduced here inside the body bacteria, the phage chromosome here would eventually try to direct or try to take over the machinery of your bacterial cell na na-infect niya, which eventually try to direct the bacterial cell here in order for that to synthesize a DNA and protein component of your phage. So gagamitin niya si bacteria since it try to take over the machinery of your bacteria. Gagamitin niya sa bacteria in order for that phage here to make its new DNA and then protein here. After na makapag-produce na siya ng kanyang mga byproducts, you try to assemble this one in order for that to make a new phage structure or particle. So after that one, you try to, as soon as meron siyang bago na phage, it's eventually try to leave the body of the bacteria by lysing the bacteria. So therefore, the bacteria here we're going to lyse. And that become here your lytic phase. Okay, now we have here your lysogenic phage, or you call that one as your temperate na phage. So the same procedure here, so the phage infect the bacteria and try to introduce its chromosome. And after that one, it try to introduce the chromosome here. The chromosome, the phage, will try to recombine with your bacterial chromosome. And as the bacterial chromosome here, try to make its own product here. So, since na naka-incorporate ng chromosome ni bacteriophage, so the product na maproproduce ng bacteria dito would eventually be under the influence of your phage. Okay, but this one, unlike in your lytic, okay, hindi siya gumagawa ng kanyang sariling na mga accessories component, but rather hinahayaan na sa bacteria mag-produce ng kanyang sariling products, but then again, that's under the influence of that bacteriophage or that phage. And later on, so pwedeng gamitin ulit sa bacteria, then try to induce here the lysis. Lalabas na siya sa bacteria. Okay, that would eventually become your lysogenic phage. Wait lang. I believe na pag lysogenic phage or lysogenic cycle here, It's the cycle here na hindi pa siya lumalabas o hindi pa niya pinapatay si bacteria na na-infect niya. Pag nag-rupture na, na-lyse na bacteria that become your lytic phase. But pag lytic fudge, ito directly, gagawa siya ng panibagong component ng fudge itself. Unlike dito, hayaan niya muna si bacteria na gagawa ng sariling products ng bacteria, but that one would have here the influence pa din ni fudge. So, hindi na ganun ka-perfect ang ginagawa ng mga products ni bacteria because again, that's been already under the influence of your bacteriophage. Example for that, we have your Corynebacterium diphtheriae. So this is a bacteria which try to cause here your diphtheria. Okay, so Corynebacterium diphtheriae are those Corynebacterium diphtheriae which are pathogenic, are those Corynebacterium diphtheriae which has been infected by your fudge. The fudge itself try to introduce here is chromosome containing here the genes we try to code here for the tox gene. This is the tox gene galing from your fudge is eventually responsible here for producing your diphtheria. So, hindi lahat ng Corynebacterium diphtheriae would eventually try to cause the diphtheria. Only those Corynebacterium diphtheriae which has been infected by your fudge are the one causing here the diphtheria. For those Corynebacterium diphtheriae which has not been infected by the fudge, they are not causing diphtheria. Okay, the next one method here of the genetic recombination is through your conjugation process. Conjugation process here is a way of transferring genetic information that may be in the form the chromosome, encoded in the chromosome, or even the plasmid through your conjugation pilus or your sex pilus. So remember that your conjugation pilus here or sex pili here is a hollow tube here that came from reproduced here by the donor and tried to having contact with the recipient. Okay, so basically what's being transferred here would be the F factor, or you call this one as the fertility factor, from the donor, and then it's transferred here to the recipient. And basically through your conjugation tube or your sex pilus. Again, you can transfer here the fertility factor encoded in a chromosome or even encoded here in the plasmid. So like for example, if the recipient were able to transfer the fertility factor by allowing it to be encoded in a chromosome, then instead of the plasmid, you call that one as the high frequency strain. Okay, then we have also here the transposon is a way also of transferring genetic information. Remember here the transposon is a jumping gene, so pwede siya mag-jump from one area to another, and therefore it could encode here allows or facilitate the transfer of the genetic information. Another thing about the bacteria, they try also to develop here their own protection against intruder or against here the incorporation of the chromosome here or genome coming from the foreign DNA. So kahit mag-transfer or mag-recombine here coming from the another source of the chromosome, that's become the foreign DNA. So pwede ni bacteria i-cut yan, tatanggalin sa kanyang genome through the restriction enzyme. Okay, so ayaw nila na ma-incorporate ka, so they could have your restriction enzyme. But of course, there's a limitation for that. At hindi natin alam what lies here with the exact mechanism for that. So we could identify here the source of the restriction enzyme. First letter of that becomes the source of your restriction enzyme. Example for that, we have the ECO RI. RI stand for your restriction enzyme. ECO stand here for your E. coli. So mean to say that enzyme has been coming from here, the E. coli as a source of the restriction enzyme. So medyo mahirap to understand here how it's able to cut on that. But this is one of their protection in order for them, the foreign here or the foreign chromosome here would eventually not able to be incorporated in their own genomic structure. Okay, so thank you.